So I'm John. I'm the NIF Radiochemistry Facility Fellow at UNSW. And today I want to briefly highlight the radiochemistry and PET capabilities in Australia with a focus on the plans for New South Wales and the current strategy to support total body PET. So UNSW is working with the University of Sydney to establish a distributed radiochemistry capability in New South Wales to support the clinical translation of radiotracers and radiopharmaceuticals towards impactful research. And to support human research utilizing total body PET, there are two main avenues that involve radiotracers. The first is the drug discovery pathway and the use of synthetic radiochemistry to develop novel radiotracers, as highlighted by Michael and Giancarlo. For example, this can involve drug design in the field of medicinal chemistry, or identifying a biomolecular target, or a process in the biomolecular application with unique properties. And then the next step is to incorporate a PET radioisotope, and radiochemical radiotracer characterization is followed. If this is successful, this can enable pilot animal imaging studies in disease models to validate initial hypotheses, and if promising, this could lead to early human research. Alternatively, the use of established radiopharmaceuticals with existing animal and human data for human imaging studies can also be used for total body PET. These radiopharmaceuticals are currently used in either early phase clinical research, they're used in later stages, or they're already used in clinical care. And in PET, the key radioisotopes are illustrated below, but the focus of this presentation will be fluorine-18 and the radiometals gallium-68, copper-64, and zirconium-89. So in New South Wales, we are establishing a distributed radiochemistry and PET capability at 3 sites across UNSW and the University of Sydney. At UNSW, radiochemistry and PET is established at the Biological Resources Imaging Laboratory, or BRIL. We have a medium-level radiation laboratory, and it's available for radiochemistry research and small animal PET-CT. At the University of Sydney, there is an existing radiochemistry laboratory at the Brain and Mind Centre, and a new radiochemistry laboratory is in the planning stages at the Royal North Shore Hospital, the site for total body PET. And across these 3 sites, radiochemistry capability will be distributed to cover synthesis hot cells, various automated synthesis modules, and quality control equipment. And more specifically at BRIL and the BMC, preclinical tools such as small animal PET-CT imaging, equipment for metabolite and stability analysis, autoradiography, and for cell and binding studies are planned to support clinical translation towards total body PET and human imaging. And as mentioned by Michael and Giancarlo, this data will allow researchers to readily progress to human imaging. So with this distributed network of radiochemistry and PET capability, we are able to provide researchers access to various radiotracers with different properties for a range of studies. And as shown in this slide, F18 and the radiometals can be radio-labeled to small molecules, small drugs, peptides, proteins, and antibodies, and even nanoparticles as examples. With F18 being quite versatile across the range of molecules, and the radiometals tailored towards the larger biomolecules. So to support preclinical and translational research towards total body PET, the 3 sites in New South Wales will have equipment for F18 radiochemistry. This consists of synthesis hot cells to provide the radiochemist or the researcher with protection from ionizing radiation. These synthesis hot cells will house automated synthesis modules that allow for remote or computerized controlled radiochemistry. And examples of these automated modules include flexible systems. These systems allow for numerous combinations of radiochemical reactions and different purification types. Another example is a microfluidic system. These systems allow for high throughput of radiochemistry reactions. And finally, cassette-based systems. And these are often found in clinical environments and they allow for the robust manufacture of radiopharmaceuticals under sterile conditions. The combination of these three types of synthesis modules will be spread across the three sites, and that will enable rapid translational radiochemistry. UNSW is currently set up for F-18 using flexible and microfluidic systems. And initially the BMC will be set up for flexible systems, and the North Shore site will be set up for cassette-based systems. And over time, we'll increase the capability and capacity of these three sites. As the radiochemistry involving radiometals is less complex than F-18 and usually involves handling lower amounts of activity, less complex equipment is used. However, the research is just as impactful. Lead-shielded workspaces can allow for radiochemists and researchers to safely handle radioactivity manually with their hands. And alternatively, compact hot cells with automated modules can also allow for completely remote syntheses like that in F-18. Manual radiochemistry for radiometals is currently available at UNSW, and it will be available at the BMC shortly. With respect to the characterization of radiotracers or quality control, we are equipped with standard analytical equipment for radiotracers. And this table shows a comprehensive radiotracer characterization for a preclinical gallium-68 labeled radiotracer that was synthesized and characterized at UNSW for small animal PET imaging. And although you don't need this level of characterization for preclinical studies, this type of characterization is found in radiopharmaceuticals used in humans. And this data allows researchers access to high quality radiochemical synthesis and quality control data, and it will allow them to progress applications towards human research in a faster manner. So to summarize this section, New South Wales will have a distributed radiochemistry and PET capability to enable drug discovery and radiochemistry development. UNSW and USID are strategically working together to ensure that the radiochemistry and PET infrastructure will allow for translational research to support human research utilizing total body PET. If drug discovery or radiochemistry development is not of interest, researchers are able to use established radiopharmaceuticals in total body PET. So established radiopharmaceuticals have existing preclinical and/or human data, and this includes information on toxicology, biodistribution, dosimetry, and safety. And they are used in human research, clinical trials, or in routine clinical care. The use of established radiopharmaceuticals can simplify human research applications for studies in total body PET. And in this case, the supply of these radiopharmaceuticals for human use, they can be accessed through commercial or licensed manufacturers or radiopharmaceuticals, or they can be accessed through generally in public hospitals. But it's also important to note that some of these established radiopharmaceuticals, particularly the ones in the early stages of human research, they may be protected by IP or require commercial and special access agreements. This table shows a list of PET radiopharmaceuticals that have been used in human research or clinically in Australia. And as you can see, we have a wide range of drugs and biomolecules labeled with various PET isotopes, including carbon-11, fluorine-18, iodine-124, and for the metals, gallium-68, copper-64, and zirconium-89. And the majority of these radiopharmaceuticals are used in oncology or neuroscience, but with the unique properties and capabilities of total body PET versus conventional PET, it'd be interesting to see if any of these existing radiopharmaceuticals can be used and applied to other fields of medicine by researchers with different areas of expertise. So to assist researchers with information on radiotracers and their availability, a radiotracer finder has been developed. This is a National Imaging Facility initiative, and a link to the website is indicated above. Perhaps Yorgos, you can place a link for our viewers to see in the chat. In the Radiotracer Finder, researchers can search the database, and for now, the database is populated with information on radiotracers available from NIF partners. Users can search the database using keywords, disease types, or radioisotope, for example. There's also general information on clinical PET units, facilities, supplies of radioisotopes, and clinical trials that are in place at the moment. Information on who to contact is also available, so I encourage you to look at the Tracer Finder to assist with your planning for research. Just to illustrate the ever-expanding PET radiochemistry capability in Australia, the supply of radiopharmaceuticals, radioisotopes, and radiochemistry facilities are available all around Australia and can support researchers interested in the field of PET radiochemistry and PET imaging. Cyclotron and PET radiochemistry facilities are available in almost every state, and this includes commercial and publicly funded suppliers. And I do want to refer you to 2 national imaging facility websites that further highlight the infrastructure and the expertise available to support researchers, particularly through the facility fellows. In terms of accessing radiopharmaceuticals and the total body PET project pipeline, access to radiopharmaceuticals should be discussed with Yorgos, the NIF Total Body PET Facility Fellow at the University of Sydney. In particular, access to radiopharmaceuticals should be discussed in the onboarding stage in relation to project feasibility, the planning stage in relation to regulatory compliance, and at the execution stage, which is the supply and the execution of the imaging study itself. And finally, for researchers who would like to access radiochemistry and PET facilities in New South Wales, the University of New South Wales and the University of Sydney, we're available to assist and advise. The facilities at UNSW are operational as we speak, and the teams at both UNSW and USyd are able to assist in each area of the drug discovery and translational research pipeline.